Chapter 3. In this chapter, we're going to be talking about observing microbes and making sure that the ways that we visualize the microbes gives us as much information as possible. The higher order learning outcomes that I'd like you guys to get to by the time we have our celebration for learning include comparing and contrasting the use of particular microscopes if given a specific situation. So I'll give you guys some examples, and you have to think about which microscope or staining technique might be best. The other thing I'd like you to know is to be able to infer what type of microbe is being viewed with a properly labeled image. So what can you tell from a picture of a microbe, and maybe what are the limits of what you know from that picture? We're going to start this chapter with an opening case study, as always. In this case study, we have two kids, ages 8 and 11, that fell ill after eating some canned chili. They showed signs of weakness on both sides of their body. Their eyes weren't tracking very well, and they were having some limited paralysis, sort of starting at the tops of their body and spreading downwards. This was pretty alarming, as you can imagine. And on further examination, it was revealed that they had eaten some canned foods that had been damaged and had been contaminated with botulism. Now, this particular bacteria, you can see from the microscope image, has club-shaped endospores on one end. And so we started this chapter with this particular case study because of those distinctive club shapes. So Clostridium botulinum, which is the bacteria that causes botulism, produces an endospore, and that's what you're seeing here on the end of these bacterial shapes. And it's very characteristically at the end of this particular bacteria. They also included this case study because these children, once they were diagnosed with botulism, they were confirmed to have botulism by looking for the toxin that botulism produces. So this particular bacteria produces a neurotoxin. So we have our picture of the brain over here. And the bacteria makes a chemical that causes paralysis throughout the body into the nervous system. And fluorescence microscopy was used to help detect the presence of that toxin. And so that was also another diagnostic way of determining whether somebody has botulism. So this case study is an important one. We think about contaminated foods like canned foods that can be contaminated with botulism. But we also know that honey is something that we can find botulism in as well. And so we are very careful not to give honey to children under 1 because they can't always report to us the signs of that early paralysis. Okay, so that was our opening case study, and right after that, as we start this chapter about observing microbes, we have another case study. So in this particular case study, we're thinking about a 28-year-old woman who came into the emergency room with some alarming features. She had a fever, she couldn't put her chin to her chest, she has a stiff neck, an intense fever, some dizziness, and was really just feeling very acutely ill. They immediately did a spinal tap and collected some cerebral spinal fluid. So again, we have our brain over here. And over here on the left-hand side, we see what typical or normal CSF fluid looks like. It should be completely clear. The blood-brain barrier that coats our brain and our nervous system, our spinal cord, is really good at preventing any kinds of contamination. But when there is an infection in this system, you can see that not only do bacteria grow in the cerebral spinal fluid, but bunches of white blood cells or immune system cells are also visible. Now, the presence of this cloudy CSF fluid is very indicative of bacterial meningitis, and this is a bacterial infection that causes severe neurological impairment and can really be dangerous in a young set. So we talk a lot about how the very young and the very old get sick, but bacterial meningitis is sort of an equal opportunity bacteria, and it can get in and cause inflammation of the meninges, which is the covering around the brain and spinal cord, and can cause blindness, deafness, and death if not treated very quickly. So we see a very healthy 28-year woman in this particular case study, and that can happen. Now, why in this chapter, when we look at that tube of cloudy CSF, we can see with our eyes that there is an infection happening, even though we may not be able to see the bacteria themselves. And that's kind of a great introduction to how we observe microbes, because sometimes we look for the presence of the microbe rather than the microbe itself. In this case, we can't really see the individual bacteria in that tube, but we know that they're there. Some other examples of that is you'll see a pile of bacteria growing on an agar plate. We call that a colony. And even though we're looking at a representative of maybe 5 to 10 million bacteria, we know that they're there even though we can't see the individual cells. Another example you might see at home is some mold on your bread. You know that there's a fungi present even though you can't see the individual cells. So that's kind of a nice introduction to how we're going to be observing microbes in many different ways. The general ranges in size of viruses, prokaryotic microbes, and eukaryotic microbes is an important thing to appreciate and understand. In this figure, they've kind of lined them all up for us. So we have, at the very smallest, we have the viruses. And viruses are the smallest types of microbes that we really look at. You could also consider prions, which are even smaller than viruses in this category. And they are going to be typically less than 1 micrometer big. We tend to measure viruses in the nanometer scale, and so that's 1,000 times smaller than the micrometer scale here. And then we have bacteria. Now bacteria can range in size, but they do tend to be smaller than our eukaryotic cells. So we talk about the range of prokaryotic or bacterial cells as between about 1/2 a micrometer all the way up to 10 micrometers in the very biggest. And then here we see a human red blood cell. So the red blood cell is the smallest cell in the body, and it ranges in about 8 micrometers. It is the smallest because it does not have a nucleus, which is an interesting fact about those types of cells. So our eukaryotic human cells range in size from about 10 micrometers to much, much larger than that. Now below that we have our paramecium, and here this guy, we're just looking at a part of this paramecium. This paramecium in total is probably 100 micrometers long, so that's 1/10 of a millimeter. So these cells are much, much larger. So eukaryotic microbes tend to range from 10 to over 100 micrometers in size. Here we can see a picture of this amoeba, and we see the outline of the amoeba, but importantly we can see the nucleus. And when you look at the images that we talk about in this chapter, see what kinds of features you can determine inside of an organism, and that gives you a lot of information about it. In each of the figures in this textbook, the size of an organism in every image is illustrated by a scale bar here in yellow. So the size of this yellow band from this side to this side is 50 micrometers in this picture. And so that's very useful in helping us understand more about an image and to see which type of microbe we might be dealing with. In black on the left-hand side of the scale bar is the type of microscope that was used. And so this side has LM, which stands for light microscope. We'll be talking a lot about the different kinds of microscopes. Look at this other picture here that shows 2 different kinds of cells. And again, here we're looking at an organism that has a nucleus. So we're looking at a eukaryotic microbe that has a long flagella. And in this image as well, we have our red blood cell. So again, that's that 10 micrometers about in diameter red blood cell that we see. Okay, so let's examine some of the different kinds of microscopes and types of microscopy that you'll see in these images. Now you'll notice most of these have the same code, LM, for light microscopy, but these images look very different. So there's ways that we can adapt and change the typical bright field or light microscope to help give us either more contrast or help our image show up better. The first microscope that we'll talk about is just your typical bright field light microscope. And this particular image has a stain. So here we'll see some type of the object appears sort of dark or stained on a bright background. Most microbes require a stain to be able to see them well with a typical bright field microscope. And that again is a limitation because there's some organisms that don't take a stain well or that are damaged by the stain, and so we have to be careful about those types. Here we have a phase contrast microscope, and this type of microscope uses sort of a cleaned up light signal to get a little bit more contrast and resolution from a typical light microscope. So the object looks transparent. It almost sort of looks 3D. It has a little bit more structure that you can see on the inside. It's important to note that these are all the same organism. So this is Giardia lamblia, which is a protozoan that causes diarrhea. Our next microscope that we're going to look at is a fluorescence microscope. And again, this is a light microscope, but it's using fluorochromes or fluorescent molecules to help label the cell. So in these images, we see a black background with a bright, colorful fluorescent signal. And this requires typically the use of either a specialized stain or an antibody that's attached to that fluorochrome to help highlight the image. So we see the outline of this green fluorescent molecule, and then they've used a red fluorescent molecule to highlight a different part of the cell. And then there are two types of electron microscopy that we'll look at. There's scanning electron microscopy, which shows the outside. In this type of microscopy, the electrons sort of bounce off the surface of the object. So we see very high resolution and detail, but we only see the outside surface of an organism. And then we have transmission electron microscopy. Here, again, we're using electrons, so we get a much, much higher resolution image. We can zoom in with incredible detail. We can see really tiny things inside the cell. Here's the nucleus. We can see the flagella base. We can see different organelles inside of our giardia. And this transmission takes a slice through, very typically like the kind of image we see with light microscopes, but here with electrons with higher resolution. And this type of microscope is the most powerful microscope that's regularly used in microbiology. Okay, so what are we seeing when we do use these microscopes? So bacteria especially come in some characteristic shapes and morphology. So morphology is just sort of how we talk about the shape of an organism. And in these pictures, I like them because you can see both the light microscope image and that scanning electron microscope. So that's the one that, of course, bounces off the surface of the microbe. So here we have the rod shape. So bacteria are typically found in two shapes, rods and sort of round balls or cocci. And then we have our spirochetes here in the middle. So rods, they can be single rods, sort of just arranged by themselves, or you can see them in these long chains where they're sort of connected together on the plane of division. And we see that again here. So we see a single rod, but we can see that these are kind of connected together, sort of like sausage links in a sausage chain. And that's the rod shape. Sometimes it's called bacillus shaped. And we have to be careful when we use the genus Bacillus to make sure that that is capitalized while the shape description is not capitalized. So Bacillus with a lowercase b refers to sort of the rod shape of a bacteria. In the middle here we see spirochete shaped, and this is again in the light microscope it's a little bit more difficult to see because it has a sort of this twisty conformation, and the light microscope is limited resolution. But if we look at the electron microscope, you can see these corkscrew-like shapes. And there are some really important pathogens that are caused by spirochetes. And it's the shape of this organism is caused by the axial filament, which we'll talk more about in Chapter 5. And then lastly, this is another one of the most common shapes of bacteria. It is a round ball or cocci shaped. And we can see that we can have these round balls shaped sort of all individually by themselves or in these long chains. We call that sort of streptococci. So streptococcus or strep is a bacteria that's arranged, it's cocci shaped but arranged in a chain. We can also see these round balls sort of clustered together like grapes, and we call that arrangement staphylococci. So staph is a common bacteria we talk about, and that's a cocci shaped bacteria but that is clustered like a group rather than in the chain. Okay, so here thinking about the shapes of these organisms, the types of microbes that we'll see, and then of course the microscopes we just talked about, let's put this all into perspective. So our human eye can see things somewhere between the 1 millimeter and 100 micron, a tenth of a millimeter range. So we can see with our eyes large microbes. But we can also see these single-celled organisms and bacteria using a light microscope. So the light microscope overlaps obviously our human eye, but goes down to about half a micrometer. So we can see bacteria, typical sized bacteria, with a light microscope. When we want to see the internal structures of the bacteria, however, so what's going on inside that bacteria or viruses, we need to use an electron microscope. So remember that the viruses have a range less than 1 micrometer. And so we really need these more powerful electron microscopes to be able to see the viruses, but also see the internal structures. So here we can see some of the organization inside this bacterial cell, and these are actually individual ribosomes that are copying RNA and DNA. or RNA into protein. So that's really, kind of the detail that we can get down to with our transmission electron microscope. It allows us to see things that are incredibly small. Now, smaller than that, on the molecular scale, we can use X-ray crystallography to get these images from X-rays that help us understand how molecules and even smaller particles are put together. Okay, so another case study. So in this case, we see a 6-year-old boy from India who has developed malaria. Now, malaria is caused by a protozoan called Plasmodium, and it's commonly found in tropical areas of the world where there's particular mosquito populations. This is the mosquito Aedes aegypti, which carries Plasmodium. These mosquitoes, of course, just like mosquitoes in our area, like to grow where there's wet or standing water environments. And so our case study takes place in India after there has been a lot of rain. This boy developed an intense fever, nausea, and headaches. The fever went away and came back in 3 days, and that's pretty characteristic of malaria. The fever is sort of relapsing and remitting. It comes back and goes away. And it can make people very, very sick for months at a time. Now, why did we put that particular case study in this chapter? Well, one of the ways that we diagnose malaria is by taking a sample of blood and looking for the effects of this particular protozoan. So this is a parasitic organism that lives inside of our red blood cells during one stage of its life cycle, and we call that the ring stage. So these are typical red blood cells. So we see our scale bar down here tells us they're about 8 micrometers, which we saw in our diagram previously. And inside some of the red blood cells, you see these Plasmodium parasites. And they characteristically form these sort of rings during this stage. These organisms will multiply and create new Plasmodium particles that will then burst out of the red blood cells, reinfect liver cells, and then can be transmitted from this patient by mosquito to another patient. So this microscopy is used to help diagnose malaria, which is why it's here. Now, these cells don't look like this just taken right out of the body. There's a particular staining technique that we do to be able to see this ring stage within the blood cells. In this case, it's called the Giemsa stain, which is pretty common for eukaryotic organisms. So staining is a process that we do to increase contrast or give us some more information about a specimen. The stains themselves, the chemicals that we use to stain, tend to be positively charged, and they are attracted to the cell membrane of different organisms because the cell membrane is negatively charged. So here, this diagram here just shows you the simple procedure for making a stained slide. So you're going to spread the bacteria on a glass slide, allow it to dry, add the dye that you're going to use in this stain. In this case, it's methylene blue. You rinse the stain off, dry it, and then view it under a microscope. Depending on the type of microscope you're going to use, you may have to put a cover slip or do other processing techniques. Now, a simple stain can tell us the size and the shape of an organism, but beyond that, the stain is not really designed to give us more information or help tell us the difference between bacteria. So if you had 2 rod-shaped bacteria on your slide, they would both be rod-shaped, and you could tell that with a simple stain, but you wouldn't be able to tell the difference between the two. To help get more information from the specimen, we tend to use differential stains. So differential stains can tell us the difference between different organisms. The most common differential stain in microbiology is the Gram stain, and so here we see a diagram of how that stain is conducted. Differential stains end up having 2 different colors at the end depending on the features of the bacteria being stained. So because of that, they may start off with the organisms being one color, and then there's a mordant or a step in the staining that helps the stain bind to one type of cell. And then there's typically a decolorizing step where you remove the stain from another type of cell. So we'll see that in this example of the Gram stain. So here we're going to put our cells on the slide and allow them to dry to the surface. So they are sort of unstained at the beginning, both gram-positive cells and gram-negative cells. The first stain that we use is called crystal violet, and it stains the cells a very dark purple color. So at this step, both types of cells are still dark purple. And then the next step is adding that mordant. In the case of gram stain, it's iodine. So iodine makes a large complex with Crestal Violet that helps it stick into the gram-positive cells. This stain that happens in gram-negative cells, even with the mordant, it will be able to be removed. Now, we do the decolorizing step right after that. And here we take alcohol and wash the stain out of the gram-negative cells, and it is trapped or is left within the gram-positive cells. So at this point, if we stopped the staining technique, we would only ever see gram-positive cells because they would be dark purple and our gram-negatives would be clear. So we come back with a counter stain that gives the negative cells a color, and in this case it's safranin, which is a light pink color. Now if you add light pink to purple, you still get a dark purple color. So these stains, these gram-positive cells, are still stained with the safranin. Their color under the microscope, however, doesn't change. Now, I said at the beginning that this is a really important stain in microbiology, and that's because the way that the stain complexes within the cell tells us something about the cell wall. So the cell wall in a gram-positive organism is made of proteins and sugars that are put together in a way that traps the iodine crystal violet complex. In gram-negative cells, the cell wall structure is made of mostly lipids, and so it's not trapped in this complex, and so when we add alcohol, it sort of washes away those extra lipids and allows the stain to be removed. So at the end of the day, not only do we see purple and pink cells, but we can tell that they have different cell walls, and many of our antibiotics and treatments rely on one type of cell wall or another, so we can make a very quick diagnostic decision about how to treat a patient based on this very simple staining technique. Here is a picture showing you an example of what those cells would look like under the microscope. So down here we see these pink cells. These are gram-negative bacteria. You can see the scale bar here. And up in this picture we see both dark purple and pink cells. So our dark purple cells are gram-positive cells, and these pink cells, you can tell, are much, much larger. These are actually human cells that are in this particular sample. So if they've scraped from a patient a particular area, they've collected some human cells along with the bacterial cells. And notice these human cells are pink, and that's because the protein sugar that we're looking for in the gram stain is not found in human eukaryotic cells. So that structure is called peptidoglycan, and it's not seen in human cells but is found in some bacterial cells. We see this dark purple stain. Some other really important differential stains that we'll learn about. One is called the acid-fast stain, and this acid-fast stain uses the same kind of steps. So it has a primary stain, it has a mordant, which is typically heat or chemicals, and then we use an acid decolorizer. So it has acid and alcohol to decolorize the cells before we stain them in a different way. So it counterstain them blue. So these acid-fast stains are used to detect Mycobacterium. Mycobacterium have a very unusual cell wall that has mycolic acids that is very difficult to penetrate with the stain. So in the first step, when we add the color to these cells, we drive it in with heat or with a very strong chemical, and the waxy cell walls are penetrated. But once you allow them to cool, even the acid alcohol can't get them out, so they're considered acid-fast. So acid-fast positive bacteria are in the Mycobacterium family because of those mycolic acids in their cell wall. This stain here, we saw a similar stain to this in our opening case study, but this is an endospore stain. So we're looking for the spore that a bacteria makes, and we stain that green. Again, we use heat as our mordant in this stain, and interestingly, water is simply used as our decolorizer because this green dye that's used is easily washed out of a regular cell, but it stays trapped within the endospore. Another important differential stain is the capsule stain. So the capsule, we'll learn about in Chapter 5, is a carbohydrate layer outside of the bacteria that helps serve as sort of an invisibility cloak. And carbohydrates are not positively or negatively charged, they're fairly inert. And so the stain itself does not stick to that sugary coating on the outside of the bacteria. So in the capsule stain, we use a stain that sticks to the bacteria, sort of positive basic stain, and we use a negative stain, a sort of an acidic stain that stains the outside or the glass slide basically. and we look to see, is there a space between the bacteria and the stain on the outside on the glass slide? And that space in between is what we call the capsule, that carbohydrate layer that does not react with the stains. So again, these differential stains all give us a little bit of extra information about the bacteria that we're studying. Okay, some other types of microscopy that we want to talk about a little bit. This one is the dark field microscope. So this is a type of light microscopy, but the direct light is blocked. And so we only see the light that's sort of reflected off of the specimen. So we see a dark field, so the background is dark, and the specimen itself is bright. So these are dark field microscopy. And this is a really great way to visualize spirochetes that are difficult to see with a traditional staining light microscopy technique. One disadvantage here is that any debris or dust on the slide also looks bright. So you can see these little specks and flecks are not bacterial cells, but just simply dust. Here is a phase contrast image, and this is a type of light microscopy where they use enhanced filters to filter the light so that the wavelengths of light are within the same phase. And what that does is it gives a really clean, crisp image without having to buy a more fancy electron microscope. So here we can see this organism, we can see the cilia on the outside, we can see some of the structures on the inside of the cell, we can see some of the vacuoles and compartments, and the image just looks a little bit more crisp. It looks sort of translucent, and that's some of the properties that we'll see with phase contrast. It increases the contrast without adding a lot of expense or fancy equipment. Okay, and here we see interference microscopy, where they look at light images to get sort of a more three-dimensional view. Okay, so another case study. So here, in this particular case study, we have a 48-year-old man that lives in Ohio. And he comes in to the doctor with some shortness of breath, some watery diarrhea. He's had a cough, and his muscles have ached. He's had nausea. He's had a bad fever. They ran some blood tests and found really high markers of inflammation in his blood. When they looked at his sample, they saw some gram-negative rods in his blood samples. And gram-negative rods can be indicative of a lot of things. So E. coli is a gram-negative rod. Salmonella is a gram-negative rod, and Legionella is a gram-negative rod. So we learn about this case study here in this chapter because we need another technique in this case to help visualize which type of bacteria this man was afflicted with. And so here we see an image of what they did to diagnose this man. So what they did was they used a fluorochrome attached to an antibody. An antibody can very specifically bind to certain types of bacteria. So they added a fluorescent dye to this antibody, and they asked the question, does this person have Legionella bacteria in the sample? If they do, the antibody binds to that bacteria and shines this bright green color we see. If they had added antibody to E. coli, we would simply just see a black background. So it turns out that this man's sample lit up or was fluorescent when they added the Legionella antibodies, and it turns out that he had a disease called Legionella, which is caused by Legionella pneumophila. It's a type of pneumonia that's found often in water pipes and air conditioners, and in this case he had been cleaning out the filter of a Whirlpool spa, and we think that that's how he had contracted this particular type of pneumonia. So we can use these techniques of microscopy to help us when a simple differential stain won't do the trick. Here's another example of using fluorescent microscopy. So here's phase contrast microscopy looking at E. coli cells, and here's that same image if you look at the fluorochrome that's been added. So we can see sort of a bright bacteria lit up with a particular color of fluorochrome on a black background when we do fluorescent microscopy. Okay, so the last type of microscopy we need to talk about is electron microscopy. And here is a picture of an electron microscope. Notice it looks very, very different than your standard light microscope. Electron microscopes bounce electrons off of their sample or through their sample rather than simply using light waves. And in order to do that, the sample has to be in a vacuum. So this big container here is a vacuum chamber that sucks out all of the air. And the sample can have no water in it. We'll learn in Chapter 4 about how cells are mostly made out of water. And so you have to replace all of the water in a sample with an organic solvent that's nonpolar. So we always have to be a little bit careful about interpreting electron microscopy images because there is a lot of processing involved. But those images are pretty cool. So electron microscopy, here we see a transmission TEM electron microscopy, and it is really the best way to visualize viruses. So here we have an HIV virus particle. We see the viral RNA as the dark black inside the virus particle. We can see the ribosomes inside the eukaryotic cell that this virus is infecting. So we can really visualize our viruses and look at more complicated intracellular processes using this advanced microscope. Notice our scale bar here is in the nanometer scale, so we can also appreciate that these microscopes are very powerful and can zoom in on very tiny things. And then lastly, we have our scanning electron microscope. These microscopes show us images where the electrons have bounced off the surface. And this can be really valuable, again, to see viruses, which are too small to be seen with a light microscope, but they can show us more about these host-pathogen interactions. How is it that a bacteria sticks to the surface of a human tissue? Or how do the flagella, how are they arranged on the outside surface of a bacteria? So we can really ask those questions about what does the outside look like. Now notice this picture, the bacteria are colored green in an electron microscope picture because the image comes simply from electrons bouncing off the surface. They are always generated in black and white. And then somebody comes in and does a Photoshop number, right, or highlights different things in the image with color. So somebody came in here and colored these bacteria green using a computer program and left the background sort of this orangey black and white color. Always be mindful that an electron microscope image has been colored. They're never colored to begin with. They don't show up that way in the microscope. But they're really beautiful, and they can really showcase what these bacteria look like and how they are arranged.